I want you to picture something in your mind's eye for a second. Imagine you're looking through a highly advanced electron microscope, like you are peering deep inside the human body.
Right. Getting right down to the cellular level.
Exactly. You see a cell. But it's not just, sitting there passively. It is glowing with this intense internal energy, and it's physically reaching out.
Like it's searching for something.
Yeah, exactly. It's extending these long, searching tentacles, just probing the biological environment around it until it finds its target, which is this jagged, chaotic-looking mutated cell, and then it just unleashes a chemical payload to completely destroy it.
Oh, man. What you're visualizing there is actually one of the most fascinating mechanisms in human biology.
Which is what exactly?
That is a natural killer cell, or, an NK cell. They are essentially the shock troops of your innate immune system.
The frontline soldiers, basically.
Right, because unlike other immune cells that need to be, specifically introduced to a pathogen before they can even recognize it, NK cells are just naturally equipped to roam the body, identify cells that have become stressed or cancerous, and just eliminate them on the spot.
Which makes them the perfect starting point for today's mission. Welcome to the deep dive, by the way. Today, we are exploring a massively promising breakthrough in oncology coming out of McGill University.
Yeah, this was just published on May 19th, 2026, right?
Yep, May 19. We're looking at a fascinating article from techno-science.net. And this deep dive is focused on how researchers have figured out a way to supercharge those tentacled natural killer cells we just talked about.
And the most compelling part of this, it isn't that they invented a new chemical weapon to pump into the body.
No, not at all. They figured out how to completely remove the biological brakes that hold our own immune system back.
It really represents a fundamental shift in how we approach the disease. For decades, the standard protocol in oncology has been to, introduce external destructive forces.
Like radiation and chemo.
Exactly. We use radiation, we use highly toxic chemotherapy, or targeted synthetic drugs. We've essentially been trying to carpet bomb the cancer.
But tumors are just notoriously adaptable.
Oh, incredibly. They mutate, they develop resistance, and they often survive the assault while the patient's healthy tissue takes just massive collateral damage.
Right. The side effects are brutal.
They are. But what this McGill research demonstrates is that, we don't necessarily need a bigger bomb. We just need to stop the tumor from disarming the soldiers we already have in our system.
Okay, so before we get into how the McGill scientists actually achieved that, I feel like we have to look at the battlefield itself.
Yeah, that's crucial.
We need to understand how cancer manages to survive in a body that already has these natural killer cells actively patrolling for threats. If we have this innate security force, why do tumors ever manage to grow in the first place?
Well, that is really the defining challenge of modern cancer research. Because in a healthy scenario, an NK cell encounters a mutated cell, recognizes it as a threat, and initiates a process called apoptosis.
That's programmed cell death, right?
Exactly. It forces the mutated cell to essentially self-destruct. But tumors, especially as they develop, they aren't just passive lumps of mutated tissue. They are highly active, complex biological ecosystems.
Right.
They construct what we call a tumor microenvironment.
Okay, let's unpack this a bit. Because in previous discussions we've had about immunotherapy, I've heard the tumor microenvironment described almost like a physical fortress, like an impenetrable titanium door that the immune cells just bounce right off of.
That's a common way to think about it, yeah.
But reading through the mechanisms in this new study, it sounds much less like a physical wall and much more like, electronic warfare.
Electronic warfare, I like that.
It's like having an elite SWAT team show up to a target, but the building is projecting a localized stealth field or a massive signal jammer.
Oh, that's a great analogy.
Right. The SWAT team is standing right in the lobby, but their radios are dead, their visors are fogged up, and the target is projecting a fake ID that says, hey, I'm a friendly civilian.
The signal jammer analogy is actually far more accurate than a physical wall, because a solid tumor secretes a very specific chemical soup. It manipulates the surrounding tissue to become hypoxic.
Meaning very low in oxygen.
Exactly, low oxygen and highly acidic. But more importantly, it pumps out these inhibitory cytokines.
And those are what, like messenger proteins?
Right, they're chemical messenger proteins that essentially broadcast a powerful stand-down or go-to-sleep signal.
So they're the actual signal jammer.
Yes. When your natural killer cells enter that specific microenvironment, Receptors on the surface of the NK cells interact with those chemical signals.
Mm.
The tumor is literally hijacking the NK cell's own internal off switch.
Wow.
Yeah, it puts the immune cell into a state of exhaustion, or what we call energy. The cancer basically gets a free pass to multiply because the security guards have been chemically sedated.
Okay, so the historical stumbling block hasn't been a lack of immune cells at all.
No, not usually.
The struggle for researchers has been trying to figure out how to keep the SWAT team's radios online and their visors clear when they walk into that jamming field. Because
Yeah.
if we just dump more immune cells into that environment, they're just going to get sedated too, right?
Exactly. You're just sending more sleeping guards into the lobby.
So we have to somehow block the fake signal. Which brings us to the core discovery from the Rosalind and Morris Goodman Cancer Institute, working with the Research Institute of the McGill University Health Center.
Right. They identified the specific biological receivers on the NK cell that were picking up the tumor's jamming signal,
Okay.
and they found that by suppressing just two specific proteins within the natural killer cell, they could completely short-circuit the tumor's ability to sedate it.
Let's get into the mechanics of that. When we say they suppress two proteins, what does that actually look like? Are these proteins basically acting as the antenna for the signal jammer?
Essentially, yes. Proteins in this context are often signaling molecules or receptors that dictate how a cell behaves.
Okay.
So when an NK cell enters the tumor microenvironment, these two specific proteins act as mediators. They receive the inhibitory signals from the tumor and translate that into the command that tells the NK cell to shut down.
I see.
And the McGill researchers discovered a way to block the function of those two proteins. The tumor is still desperately broadcasting its stand-down signal.
But the NK cell is basically deaf to it.
Exactly. It simply doesn't have the functional proteins required to hear it or process it. The jammer is ignored.
That's incredible.
It is. The NK cell remains fully active, recognizes the cancer, and regains its natural killing power right in the heart of the tumor.
And the results from the preclinical trials on this are staggering, mainly because of the specific enemies they chose to test this on. I mean, they didn't go after easy targets to get a quick win.
Not at all.
They tested this method on some of the most stubborn, notoriously difficult to treat tumors on the planet. The study highlights human cancer cells in animal models for leukemia, glioblastoma, kidney cancer, and triple-negative breast cancer.
The inclusion of glioblastoma and triple-negative breast cancer is particularly vital for proving this concept.
Because they're so hard to treat.
Exactly. Glioblastoma is an incredibly aggressive brain cancer. And the brain itself has an extremely unique, highly protective environment that naturally limits immune responses to prevent swelling.
Right, because you don't want inflammation in the brain.
Exactly. So overcoming the suppressive environment of a glioblastoma is arguably one of the hardest tasks in oncology.
And then there's triple-negative breast cancer, which is famous for being evasive because it lacks the three common receptors, right? Like estrogen, progesterone, and the HER2 protein that standard breast cancer drugs usually target.
Right, it's missing the targets.
Yeah, so if those receptors aren't there, traditional targeted therapies have absolutely nothing to lock onto. It's essentially invisible to our best synthetic drugs.
Which is why flipping the script to rely on the innate immune system is so powerful. Because the NK cell, it doesn't need to look for an estrogen receptor to know a cell is cancerous.
Yeah, it just knows.
Yes, it just needs to not be sedated. By proving that suppressing these two proteins works across these vastly different, highly suppressive cancers, the researchers demonstrated that this isn't just, a niche treatment for one specific mutation.
It's a broader solution.
Right. What's fascinating here is that it is a broad spectrum strategy for overcoming the tumor microenvironment itself.
There is a really powerful quote in the source material from the lead author, Michel L. Tremblay, who is a distinguished James McGill professor. He points out that this approach is phenomenally promising for patients who have exhausted all standard treatments.
Yes, the patients who are basically out of options.
Exactly. The tumor didn't respond to chemo, it survived the radiation, it doesn't have the receptors for targeted drugs. To be able to tell those patients that we might be able to simply upgrade their own internal security force to finish the job, it's incredible.
It shifts the focus of treatment entirely. But, anytime we start talking about upgrading immune cells, altering proteins, or modifying how the body responds at a cellular level, we run directly into the issue of safety.
Right, because you don't want the immune system turning on you.
Exactly. Historically, modifying the immune system has been a high-wire act with some severe and occasionally fatal consequences.
That actually brings up a critical distinction in this research. Whenever I hear about altering an immune cell's behavior, I immediately think of gene editing.
Like CAR T therapy.
Right, things like CAR T cell therapy, where scientists literally rewrite the DNA of a patient's immune cells to make them aggressively hunt cancer. But this McGill breakthrough specifically avoids permanent gene editing. Instead, they are using small molecule drugs to suppress those two proteins.
The distinction between permanent gene editing and small molecule drugs is really the crux of why this new approach is so viable.
How so?
Well, with gene editing, you are permanently altering the genetic code of a living cell. You take it out of the body, use a viral vector to insert new instructions into its DNA, and put it back. The problem is, once you create a permanently hyper-aggressive immune cell that ignores all stand-down signals, you can't easily turn it off.
Right, because if that permanently altered cell finishes killing the tumor, but then decides a healthy organ looks a little suspicious, it won't listen to the healthy organ's stand-down signals either. It could just keep attacking.
Precisely. That leads to severe toxicities, like cytokine release syndrome or autoimmune cascades, where the immune system just aggressively attacks the patient's own healthy tissue.
And it's unmanageable because the edit is permanent.
It's an unmanageable side effect, yes. But the McGill team sidestepped this entirely by using small molecule drugs. Small molecules are exactly what they sound like. They're tiny chemical compounds with a very low molecular weight.
So they can get right in there.
Right. They are small enough to easily slip right through a cell's outer lipid membrane, get inside, and temporarily bind to a specific target. In this case, are two problematic proteins.
Okay, here's where it gets really interesting, but also a bit confusing for me. So instead of rewriting the cell's underlying operating system permanently, these small molecules act more like a temporary software patch.
That's a good way to phrase it.
Or, going back to our SWAT team, it's like handing the officers a specialized tactical visor that filters out the tumor's jamming signal.
Right, right.
They wear the visor, they breach the room, they clear the target, but eventually the visor's battery dies, right? The small molecule drug washes out of the system, and the NK cell naturally reverts back to its normal, unaltered state.
Exactly. It provides the overwhelming efficacy of a gene-edited cell, but with a highly manageable, reversible safety profile. You don't leave a rogue army of permanently altered cells circulating in the patient's bloodstream.
Okay, I follow the safety logic completely, but playing devil's advocate for a second.
Go for it.
Is a temporary software patch, or like a temporary shot of espresso, really enough? If we are talking about fighting a massive, deeply entrenched glioblastoma that has been growing and fortifying its microenvironment for months or even years, how does a temporary boost win the war?
It's a fair question.
I mean, if the drug washes out and the NK cell goes back to normal, won't the tumor just start jamming the signal again and grow back? It feels like you'd want the immune cells permanently on high alert to ensure every last cancer cell is gone.
It does seem counterintuitive, I grant you that. But a temporary breach is often all the immune system needs to trigger a permanent victory.
Really?
It's due to a biological process called the antigen cascade, or epitope spreading. You have to remember that the tumor's greatest defense is its stealth field. It survives by remaining unrecognized.
So it's hiding in plain sight.
Yes. When the small molecule drugs temporarily block those two proteins, the NK cells suddenly see the tumor clearly. So they attack and destroy the outer layers of the cancer. When those cancer cells die, they literally burst open, spilling all their internal highly mutated proteins, their antigens, into the surrounding bloodstream.
Ah, so they blow the tumor's cover entirely.
Exactly. The destruction caused by that temporary strike team acts like a massive alarm bell for the rest of the immune system. All of a sudden, the patient's dendritic cells and T cells,
Which are like the heavy artillery, right?
right, they make up the adaptive side of the immune system. They detect this massive spill of mutated proteins. They realize there is an active threat. They learn exactly what the cancer looks like based on the spilled debris, and they rush the site to finish the job.
Oh, wow.
So the temporary stimulation of the NK cells is really just the spark. Once the tumor microenvironment is compromised and cancer cells start bursting, the body's natural immune cascade takes over and finishes clearing the disease.
That is deeply fascinating. The temporary patch just gets the door open so the rest of the body can swarm the room.
Precisely.
It solves the safety issue of permanent gene therapy while still dealing a lethal blow to the cancer. But, even if we have a treatment that is biologically safe and effective, it hits a massive real-world wall if it takes too long to produce or costs too much.
Oh, absolutely.
Which leads us into the logistical side of this research, which might honestly be just as revolutionary as the biological mechanism itself.
Logistical barriers are truly the silent killers in advanced clinical medicine. I mean, you can cure a tumor perfectly in a petri dish or an animal model, but if the manufacturing process is so complex that you can't deliver it to a patient at scale, it remains a laboratory curiosity.
And the current logistical state of cellular immunotherapy is just brutal. For most current therapies, doctors rely on an autologous process.
Which means using the patient's own cells.
Right. They have to extract a specific patient's immune cells through this long blood filtration process. Then they freeze those cells, ship them to a hyper-specialized manufacturing facility, spend weeks genetically engineering and multiplying them, and then ship them back to the hospital to be reinfused into that single patient.
It's incredibly labor-intensive.
It is an entirely custom, bespoke process for every single person.
And the financial cost of that is staggering, frequently running into the hundreds of thousands of dollars per patient just for the manufacturing alone.
Yeah.
But the cost in time is even worse. The entire process can take 3 to 6 weeks. When a patient is facing an aggressive, late-stage leukemia or a fast-moving brain tumor, they simply do not have 6 weeks to wait.
The disease is moving faster than the bespoke manufacturing process can handle.
Exactly.
But the McGill team completely bypassed that bottleneck. The research led by the Cell Therapy Lab at the Research Institute of the McGill University Health Center, specifically under Pierre Laineville and Linda Peltier, they found a way to skip the extraction entirely.
This is a huge deal.
Yeah, they aren't waiting to pull sick, exhausted immune cells out of the patient. They are using natural killer cells isolated from umbilical cord blood donations.
Moving from an autologous process, using the patient's own cells, to an allogeneic process, which means using donor cells, is a monumental shift. And umbilical cord blood is the perfect vehicle for this.
Why cord blood specifically?
Because cord blood is incredibly rich in natural killer cells.
Sure.
But more importantly, those cells are immunologically naive. They are a pristine blank slate.
Meaning they're much less likely to trigger a massive immune rejection or graft versus host disease when put into a random patient, right? Because they haven't learned to attack foreign tissue yet.
Exactly. Adult donor cells have decades of biological programming and memory. If you put adult donor immune cells into a patient, those cells will immediately recognize the patient's body as foreign and attack it.
Which is obviously bad.
Very bad. But cord blood NK cells don't have that aggressive memory. They can be introduced into a patient's system safely, where they will focus their innate killing power strictly on the stressed and mutated cancer cells.
The lead researcher on this specific part of the project, Chuhan Feng, noted that skipping the complex personalization process makes this therapy faster, safer, and more affordable.
It really does.
They can take these donated umbilical cord blood cells, which are often just discarded as medical waste after a birth anyway, isolate the pristine NK cells, apply the small molecule drugs to suppress those two proteins, multiply them, and freeze them in massive batches.
It essentially creates a universal inventory. If we connect this to the bigger picture, we're looking at the transition from highly restrictive, bespoke medicine to an off-the-shelf bank therapy.
Off-the-shelf cancer therapy. That's wild.
It is. A clinician could diagnose a patient with a rapidly advancing tumor on a Tuesday, walk over to a specialized freezer, pull a vial of these supercharged, ready-to-use natural killer cells, and begin the infusion on Wednesday.
It essentially democratizes what is arguably the most advanced cancer treatment on the planet. Instead of restricting this to a tiny fraction of patients who can afford a $1 million custom cell therapy at an elite research hospital, you can package this, ship it globally, and administer it in standard oncology clinics.
You eliminate the waiting period entirely.
Yeah, you slash the manufacturing cost and you hit the cancer instantly. So looking at the timeline, where does this research actually go from here? When is this happening?
Well, the underlying mechanisms are scientifically sound, but the immediate next steps require rigorous human validation.
Right, clinical trials.
Yes. The team is currently pending the necessary funding and regulatory approvals to move into their first human clinical trials. And they have strategically selected acute myeloid leukemia, or AML, as their first target.
That makes perfect sense biologically. I mean, AML is an aggressive cancer of the blood and bone marrow. It moves incredibly fast, making it the perfect proving ground for a therapy that prides itself on being instantly available off the shelf. And because it's a blood cancer, the intravenously infused NK cells will immediately come into contact with the cancer cells circulating in the bloodstream without having to navigate deep into solid tissue right away.
It's a very smart first target. If they can establish a strong safety profile and prove efficacy in humans against an aggressive blood cancer like AML, it clears the path to expand clinical trials into the highly suppressive solid tumors we discussed earlier, like glioblastoma and triple-negative breast cancer.
The global oncology community is going to be tracking these trials very closely, I'm sure.
Oh, without a doubt.
So, taking a step back to look at the entire scope of this McGill breakthrough, we've covered tumor microenvironments acting as signal jammers, temporary molecular software patches, the cascade effect of an immune response, and creating a universal bank of pristine cord blood cells.
It's a lot to take in.
It is. So, what does this all mean for you, the listener? What is the overarching takeaway here for anyone trying to understand the trajectory of modern medicine?
I think the defining takeaway is that we are witnessing a profound philosophical shift in how we treat human disease. For over a century, our primary instinct has been brute force.
The carnet bombing approach.
Right. We viewed cancer as a foreign invader that required us to indiscriminately poison the body in the hopes that the cancer would die just before the patient did. But this research proves that we are entering a much more elegant, sophisticated era.
How so?
We are learning how to decode the biological communication between a tumor and the immune system. We're moving away from carpet bombing and moving toward intelligently, precisely, and temporarily uncuffing our own biology so it can do what millions of years of evolution designed it to do.
It's working in harmony with the system rather than trying to overpower it.
It is the ultimate realization of personalized medicine, ironically achieved through a universal off-the-shelf product.
That is perfectly said. And that leaves us with one final, incredibly provocative thought to consider. We started this deep dive by having you visualize that glowing natural killer cell extending its biological tentacles to destroy a tumor. But let's build on everything we've unpacked today about how this therapy is actually manufactured and delivered.
It really forces us to entirely reevaluate what a cancer diagnosis might mean in the near future.
Right, because if medical science can successfully isolate universal, pristine immune cells from something as natural and routinely discarded as umbilical cord blood,
Yeah.
and if we can use small molecule drugs to temporarily patch those cells so they completely ignore the evasive stealth fields of a tumor, are we approaching a reality where a notoriously deadly cancer diagnosis simply loses its terror?
That's a beautiful thought.
Imagine a near future where treating something as devastating as a glioblastoma doesn't mean months of toxic chemotherapy. It's as simple as your doctor pulling a ready-made, off-the-shelf biological software update from a freezer.
An update that you receive through a standard IV.
Exactly. An IV which instantly bypasses the invisible shields around your cancer, sparks a massive natural immune response to clear the disease, and then completely safely washes out of your system without a trace once the job is done.
No permanent genetic alterations, no weeks of agonizing waiting for custom manufacturing. Just a precise, temporary, devastatingly effective boost to your own internal defenses.
The ultimate signal jammer, finally taken offline. It is a brilliant, highly realistic glimpse into what the true future of healing will look like.